Everybody, I welcome all back to our class. Today we are doing the Spiroplasma and Viroid disease. I already did the virus disease and plant disease control. If anyone having any problem, please contact me. I hope you all understand it. So today I will start the very important disease, that is Spiroplasma and Viroids. So be with me, be indoors, be safe. So I will write back in a moment. Stay. Morning everybody. I welcome you all for this special session which deals with the diseases of Spiroplasma and Viroids. So today we will do the Spiroplasma disease, which is caused by the gram-positive bacteria which lack cell walls and bound by the triple layered unit membrane. Spiroplasma has a fried egg colony with helical morphology. The cells are pleomorphic, that is, it varies from spherical to ovoid to helical and reproduces a binary fission. It is found in the phloem cells with diameter of 100-250 nm, branched with no flagella. Right? So Spiroplasma itself explains its characters. S means spherical, P means positive, O means ovoid. It's shaped from spherical to ovoid, and it gives rise to a fried egg colony. When we put egg on a pan, nowadays we are making a lot of egg because we are free. So when it put in a pan, it gives like a fried egg. That egg at start like this. It gives the shape called fried egg colony in case of Spiroplasma. And then they are basically present in the phloem cells. This is the mode of transmission. So Spiroplasma itself tells the characters, word Spiroplasma, of the characters of the Spiroplasma. The first disease we are doing is the Spiroplasma disease is the stubborn disease of citrus, and this stubborn disease is caused by the Spiroplasma citri, right? So this is also called citrus stubborn disease, that is CST, right? And it is known by different names in different countries, whereas in Israel it is known as the little leaf disease, right? So the first symptoms which started in it are the symptoms on the leaf. They are small size, right? Leaves are small size with upright position. And these leaves which are small size with upright position, they showed mottled appearance and venal chlorosis. See, if this is the shape of the seed, so first it shows the venal chlorosis. So means the chlorosis started at the veins, right? And then there are spots, yellow spots coming like this, right? And these spots come at random and it gives like a yellow, green, yellow. It is mosaic pattern. We call it mottling, right? So it results in the shortened stem internodes. Shortened stem internodes like this. These are the internodes, and these are shortened. And these shortened internodes leading to the bunch-like growth. See, they bunch, right? So leading to the bunch-type growth, trees affected while young. When these trees are affected when they are young, they show stunted growth. They're smaller. Like the normal trees are bigger, right? So they are smaller, stunted, right? And then we are coming to the then there is I already told you, the leaves are shorter, the little leaf, chlorosis. Then we have chlorosis, which is every time followed by the necrosis, and then there is a stunted growth and leaf defoliations, right? So when the shoots are bunched, as in time of Holi and Diwali, we will see people wearing like this headgears, right? They are red in color sometimes. And when they are bunched and these leaves are like pointed this, sometimes spindle shaped, and they look like a chodail, that is witches, guru. So the shoots abnormally bunched and they form a witch's broom. So then we are coming to the next is the fruits. Fruits are basically small size, right? And these fruit are immature, acorn shaped. Acorn means deformed, not a balanced shape, right? And these fruits, they have very poorly developed seeds. And these fruit drop is very common seen in these, severely affect fruit trees. They have very bitter flavored. We can say bitter flavor, or we can say one thing, insipid, INSIPAD, insipid, right? So then we are coming to the control. Control is that as prevention is better than control. First is rogging, that is a removal of the infected trees. The trees which are infected with Spiroplasma, they should be removed. And the root stocks which are taken not from these infected trees, they are taken from the healthy trees. Spiroplasma free certification should be there so that the farmer can know that these trees are not affected. And the last is the elimination of reservoir host. There is one reservoir host called Tripsacum. So Tripsacum should be removed so that na rahega baanch, na bajegi bansuri. So this reservoir host should be removed, so it will give us full protection for the disease. So now the second disease we are coming is the corn stunt. Corn stunt, as the earlier we had the stub-borne disease caused by Spiroplasma citri, and this is caused by the Spiroplasma kunklei. So kunklei is a disease which results in the first small chlorotic stripes. These are the small chlorotic stripes. This is a disease is corn stunt. So corn stunt results in the small chlorotic stripes. So this basically most economically important disease of maize in America and Mexico, and we know we did in BSc also. It might there is a video is also present. So maize is basically grown. 85% of maize is grown in the corn belt region of US. You can see that. Refer to that video also, right? So the first symptoms which started is the chlorosis of leaf margins. See this. So the first symptoms are the chlorosis of the leaf margins, like this. Chlorosis of the leaf margin, which is the first symptom we are dealing with it. And then it followed by the reddening of tips of the older leaves. Then the leaves, as chlorosis of leaf margin started, then it result in the reddening of the leaf tips, right? So these are the two characteristic symptoms. Then we explain the small chlorotic spots appear at the base of the new leaves. See, small chlorotic spots at the base of the leaves. Then which coalesce means fuse to form stripes, and these stripes extend towards the, and these coalesce, and these form a stripes, and these stripes moves towards the tip of the leaves. These are yellow stripes, and these stripes moves towards the tip of the leaves, and these stripes they alternate with the green background. Green background means chlorophyll. So because of this it gives the mosaic pattern, right? So as we have just seen in the leaves, right? Plants are stunted because there is a chlorosis. Chlorosis is written by necrosis. Then less photosynthesis. As we know, the photosynthesis is inversely proportional to the respiration. So respiration increases, so it results in the stunted growth. So stunted growth, defoliation, we did already these things, right? So then plants are stunted and numerous tillers developed at leaf axils. From the leaf axils, number of tillers rises, and base of the plant, giving it a bushy appearance. So in the leaf axils, number of tillers were developed, not from the leaf axil but from the base of the plants, giving it a bushy appearance. The infected plants have shorter internodes, which I already told you, and they have secondary shoots. Means they say a lot of secondary shoots and even from the base, and this give rise to a bushy appearance, right? So this is the second disease we are doing. So now we are coming to the third disease of it, that is the this one, Opuntia witch's broom. Here very important, same as that extensive chlorosis. We know we have chlorosis, and then chlorosis, this is chlorosis, then diebacks of stem. The stem will die back, and chlorotic circles. And we know that chlorosis, if chlorosis occur, it is every time followed by the necrosis, and that result in the defoliation and death, then death. So simple, that is the first is the chlorosis, diebacks of stem, chlorotic circles, rings and spots on the cactus. So proliferation of same as we have did last time, that is axillary shoots. It is also in the other disease which I am not covering, Bermuda grass white, the symptoms are same. But here the proliferation of axillary shoot is prominent in Bermuda grass white. So leaves are small and it results in the death of the plant. The disease cycle, it inhibits the basically the phloem sieve tube of host, and vector is transmitted by the leaf hoopers. So in both here also inhibits in phloem, same, the disease cycle is same. That's why I am not taken earlier. So the control is also same, removal of infected trees, which we did in the stub-borne disease, elimination of reservoir host. So seed symptom of Spiroplasma free certification, that it is free of the disease. So these are the important disease I have taken, and these are the symptoms I explained you. So please go through these symptoms and get back to me any problem you have. Thank you. Morning. I welcome you back for the viral disease session. We did the Spiroplasma diseases, and I hope you understand all the diseases very clearly. If any problem, you can contact me. Right now, I am doing the viroid diseases. So viroids are the small, low molecular weight RNA molecules that can infect plants. Viroids lack protein coat, circular, single-stranded RNA, brackets naked, with 359 nucleotides in, we will do the disease, that is potato spindle tuber viroid. And it is 371 nucleotide in Citrus exotis, the disease I am taking after that right now. So viroids are the smallest known agents of infectious disease, transmitted generally through the mechanical means. The common symptoms of viroids are chlorosis, vein clearing, necrosis, venal discoloration, stunted growth, epinesty. Epinesty is a very important symptom, particularly in viroids, and death of host. So when we, we already told you, in Spiroplasma, the viroids did the same thing. So in case of viroids, so see, R RNA, D venal discoloration, and venal discoloration, E is epinesty, and S is the smallest known pathogens, and it lacks the protein coat. So viroids itself tells us that it is a RNA, it causes venal discoloration, and it causes epinesty, smallest known pathogen, and lacks a protein coat. And the first disease we are taking is the Citrus exotis. Citrus exotis, which is caused by the Citrus exotis viral disease. So it is having first symptoms Citrus exotis is showing is on the leaves. The leaves become yellowish purple, right? Curled and twisted, right? See, if this is the leaf of the citrus, first it shows the color change, that is yellowish purple, and then it is twisted. inside and this side, upside and inside. Twisted, and as it twisted, you understand the surface area decreases, and the surface area decreases, so photosynthesis is reduced. As we know, respiration increases, so it results in the first chlorosis, then necrosis. And the characteristic feature it shows is the first is the stem shows yellow blotches after leaves, and stem and leaf shows epinesty. I already told you viroids, the characteristic feature is the epinesty. And then when it roll insets, it gives a voice of cracking, and then it results in the darkening. See, this darkening of the midrib. Whereas in this we have the in case of stubborn disease, darkening of the veins, but in case of here it is a darkening of the midrib. See, right? Next, then there is we will see plants, as we said, that chlorosis followed by necrosis, then we have the stunted growth because there is a defoliation of leaves, and as there is no food formation, the main is citrus economical concern is the fruits, that is it shows stunted growth. So besides this, it shows vertical split in the stem like this, right? And the fruits, they are having a stunted, small, less yield. So these are the characteristic features of this. So transmission here is by means of a grafting or by knives, budding knives. So it is also transmitted by dodder. Virus enters through phloem elements, same as that of Spiroplasma, and the control is use of healthy seedlings. And the tools which are to be used like knives, they should be infected with the 10-20% solution of sodium hypochlorite. And the last, that is the removal, and that is rogging, that is removal and destruction of the virus infected plants. So this we did the citrus exotis. Now we are coming to the next disease, that is the kadang disease of coconut. And this kadang disease of coconut is basically caused by the CCCVD. Okay? Kadang coconut disease, viral disease. And these, they show bright yellow spots. And bright yellow spots, it did the same thing like chlorosis followed by necrosis, then the defoliation of leaves. So then if defoliation of leaves, the photosynthesis decreases. So number and size of leaves also reduces because it leaves curls, and it results in the death of the growing bud and inflorescence. So if the inflorescence is not there, then we don't have the fruiting. So transmission is same as that is the grafting and budding and through dodder, and the control is same. Healthy seedling, disinfectant with the of tools with the sodium hypochlorite 10-20%. So we came to conclusion, the crux is abstract is that the citrus exotis and kadang, they have the symptoms exactly the same, except the vertical splits which we will see there, and particularly the epinesty. So then we are coming to the third, that is called potato spindle tuberoid viroid. It is called PSTV. This is the first viroid to be identified. Please mind it, this is for the UGC, you will get this particular. And the first potato spindle tuber viroid is the first viroid to be identified, and it is single-stranded RNA. The leaves, they become yellow or purple. Mind it, it is yellow or purple, curled and twisted, same that as we did in the citrus exotis. It results in the smaller leaves, right? Because they curl, the size surface area decreases, and they shows the spindle like elongation. So in abstract we say chlorosis as we did in kadang followed by necrosis, but here change, and here the leaves, it results in the after chlorosis necrosis. So we have the leaves reddening and purpling. It is in case of this. So one change which we will notice from Kadang, here the chlorosis followed by necrosis, but then there is a reddening and purpling of the leaves. So these are the basic characteristic position of this. Here we will again see the rolling and twisting of the leaves, which I already told you. The leaves, it will roll and twist, right? And the leaves are small, erect, potato plants erect, dwarfed with stunted growth, because as leaves they roll inside, the photosynthesis stops. So the plants, they show the stunted growth, and then that's why they are dwarfed. As I told you that photosynthesis is inversely proportional to the respiration. Tubers are elongated with tapering ends. So as we see, I already told you, here the midrib increases and then tubers are elongated. This elongated with this tapering ends, right? So tubers are elongated with tapering ends, and these plants have a bunchy top-like appearance. So because internodes are shortened up, and the diseased plants, they show the bunchy-like top appearance. The transmission is same, that is mechanical transmission knives, which are used to cut the seed tubers, right? So seed tubers I already told you last time also. So when this is potato and this is central bud, and these are the axillary bud, and this is the central bud, this is the axillary bud, and this is called node, and this is called internode. And when this is mother axis and we cut it on the right angle, which consists of two internodes and one node, is called seed piece. So this is the seed piece which I am talking about. So then the knives which you use to cut these seed pieces, seed tubers or seed pieces, it should be disinfected with the sodium hypochlorite, so that there is no transmission of the viroid to the healthy one. The viroids also transmitted by the insects, and the main which we left is the rogging, that is the removal of the infected plants and burning. The disease-free potato tubers should be used. Washing of hand and sterilizing. Washing of hands is also required. Nowadays we also do the washing of hands. So it is a characteristic feature nowadays. So we wash hand. So in this also we wash hand to prevent viroids. And viroid and virus, they are very close friends. So they go hand in hand. So today the basically abstract is remain indoors, remain safe. I will contact you again for the next topic. Thank you. Bye bye.